Hi, everyone. It's my pleasure to continue this talk. And now I would like to switch gears from nanotechnology and basic description what nanotechnology is to actually the ability to make nanotechnology safer, more sustainable, and talk about nanotoxicology. My name is Olga Tsyusko, and I am Associate Professor at Plant and Soil Sciences Department at University of Kentucky. So with that, as you already heard from Paul a lot about applications of nanoparticles in different areas of life, right? In different industries, cosmetics, food, in medicine, in research, in environment. And for example, on the right here, you can see examples of applications of nanotechnology in agriculture. In agriculture, we're really striving to improve the ability of, for example, plants against the stress. You can use nanoparticles. You can use nanotechnology to enhance the plant growth and thus to increase the crop yield. Or you can use different nanosensors for observations of plants when they are growing. Also, it's possible to use nano as nanopesticides, but in all of those applications, we often achieve the benefits of nano, such as in nanopesticides can be used directly against different pests, right? So it does kill the pest. But what about non-targeted toxicity? What do I mean by non-targeted toxicity? It's toxicity to non-target organisms. So if those nanopesticides is targeted pests, but they are not supposed to be targeted, like, for example, earthworms or bees or aquatic organisms, if there is sunrunoff, if there is runoff of those nanomaterials into surface water. And here is a graph that shows the number of publications in the last decade, basically, that focus on nanopesticides, right, and development of nanopesticides, testing nanopesticides, and the one in red and green, really small tiny bars, is the publications that actually take into consideration also assessment of risk to non-targeted toxicity, non-targeted organisms. So also what is important that during the life cycle of nanomaterial containing products, there is unintentional and intentional release of nanomaterials into the environment. For example, during use, during production, a lot of them nanomaterials will enter wastewater treatment plant, right? Will enter wastewaters. Wastewater is an environment that is very rich with sulfur and phosphorus. And then from there, they will be deposited into soil. For example, in US and Europe, more than 50% of biosolids or sewage sludge that generated through wastewater treatment process is applied to agricultural soils as fertilizer. And here we are, the nanoparticles will deposit it in soil where they will interact, right, with the different microorganisms, and also can be taken by plants, eventually by animals through food chain, and can reach to people, to humans. Also, nanoparticles can be deposited in landfill, and part of it may be incinerated, and that we have particulate nanoparticles available for inhalation in the air. So what happens with toxicity? As you already heard from Paul, that there is a lot of different nanomaterials available with different coatings and shapes and sizes. And obviously we need to have some kind of rapid screening method. So high throughput screening can be available by using screening through, for example, single cells, right? So it's in vitro screening, and you can screen for cell viability, or if there are cells that carry a specific reporter, like green fluorescent protein reporter gene, right, that you can look at the change in fluorescence intensity. There are also other assays can be used for generations of reactive oxygen species, and that's in vitro. And obviously, in vitro is a good fast screening technique to really estimate if these nanoparticles even safe. But in vitro toxicity not necessarily will translate into in vivo, which is whole organism toxicity. And that's why we definitely need to test also using the whole organism. In this case, for example, whole organism is zebrafish. And you can look at the hatching, for example, and different morphological abnormalities. Again, there is green fluorescent protein reporter genes you can also use. So overall, nanotoxicology is basically a study of toxicity of nanoparticles. And this is necessary so we can predict and eventually evaluate risks from exposure to those nanoparticles, both in environmental and human health. So what makes model organism is really powerful organism for nanotoxicity. And this is just example of one of the model organisms that my lab works with frequently, and I really enjoy working with it. It's Cyanaraptitis elegans or soil nematode. So soil nematode is really small, 1 millimeter in length, right? And it has very rapid life cycle from the egg to the adults, take only 3 days. Also, the beauty of it, the genetics is known. It's sequenced, annotated, and it also has a lot of different mutant strains and green fluorescent protein reporter strains that can be used. So endpoints that we estimate in C. elegans, we start with mortality, which is least sensitive endpoint. And if you see any mortality due to exposure to low doses of nanoparticles, that's definitely suggests that they are pretty toxic. The least, the less sensitive endpoints are reproduction, locomotion, and then you can go more into mechanism and look at which pathways, biochemical, biological pathways are affected. Also, such endpoint as production of reactive oxygen species and various molecular biomarkers that will be indicative of stress induced by exposure. So when organism is exposed to the nanoparticles, for example, nanoparticles will be ingested, right, and will accumulate in the intestine, and from intestine they will be absorbed. So when nanoparticles enter the environments rich with proteins, what happens depending on nanoparticle surface chemistry, such as coating, it will attract different protein to bind to the nanoparticle surface. And in fact, if there are different, for example, charges, right, there will be different proteins attracted to that surface. So the affinity for the binding of proteins differ depending on that surface chemistry. And what happens with the nanoparticles? So they are being taken into the cells, one of the main route entry of the cells through endocytosis. So forming those kind of like vesicles, right? Eventually those nanomaterials will enter into lysosomes through endocytosis, and lysosomes are very acidic rich environment, like it's very low pH. So the nanomaterials, if they're not stable, they will be dissolving and releasing ions. Also, depending on which nanoparticles we're dealing with, if it's, for example, silver or zinc oxide, they prone dissolution. So the dissolution can occur before entering the cells or even after entering the cells. So the dissolved particles here are shown in those blue circles. After they enter the cells, if it's particle, right, not dissolved, then we have protein corona forming around those particles. And when proteins actually bind to the surface, they can get denatured, and we end up with a lot of misfolded proteins, which as a result induce endoplasmic reticulum stress, unfolded protein response, can result in apoptosis, cell death. Also, nanoparticles or ions can induce production of reactive oxygen species, cause mitochondrial dysfunction, and activate cascade of events that lead to genotoxic events, right, such as DNA damage, and again will result in apoptosis. So obviously, the nanoparticle properties are really important in determining toxicity. So if you look here on the right, and I know Paul already mentioned some of those properties, and such as size of nanoparticles, solubility, surface chemistry, shape, concentration, chemical composition, all those important on how they affect toxicity. And also how it affects nanoparticles uptakes or bioavailability, bioaccumulation, and which responses will be generated in response to such exposure. One of the properties that I would like to emphasize that especially important, critical, one of the most critical ones for unstable nanoparticles that are prone to dissolution is solubility. So in this diagram, actually, you can see examples for toxicity of silver nanoparticles and copper oxide nanoparticles. When we are working with nanoparticles that are unstable, we also require to include the treatment with their ions. So ionic treatment would be silver nitrate, for example, silver salt, and also copper sulfate, for example, copper salt, right? So we have ionic treatment, so we can tell how much toxicity can be produced due to dissolution. So if you look, those are model organisms on the right here that have been tested, and the toxicity levels can be divided by extremely toxic, very toxic, toxic, harmful, and those are non-classified. And for silver nanoparticles, for example, such aquatic organisms as Daphnia, algae, fish, the nanoparticle toxicity is silver nanoparticle toxicity is from extremely toxic to very toxic. If you look down here for silver ions, the toxicity is even higher, right? So lower concentration of silver causing mortality, for example, in those same organisms. So what happens when we have exposure to silver nanoparticles? They also release silver ions, which cause this toxicity at the really small concentration. Plus, if they did not dissolve, then we have particle-specific toxicity due to unique properties of nanoparticles. So that's important to take into account. With copper oxide, again, we have toxicity for nanoparticles in toxic area for algae and Daphnia. And for copper ions, the toxicity is higher. Again, they also rapidly dissolve and release copper ions. And depending on the media in which organisms, such as nematode, for example, being tested, the toxicity level can be changed. And in our case, in our experiment, we use different media in this study, and the toxicity actually is moving to extremely toxic and very toxic area for particles. So also another properties that are really important for determining toxicity is size, shape, and coatings. So this is example, this is for mammalian organisms, for mice, and depending on different size of the particles those organisms are being exposed to, the accumulation is different in different organs. For example, at more than 150 nanometers size, accumulation is higher in lungs, liver, and spleen. But if you go below 5 nanometers, then you see that it's accumulated in kidneys, right? And none of the other sizes are accumulating in kidneys. You can see it here. So what happens when nanoparticles decrease 6 nanometers in size and the diameter, and it's for gold particles in this case, they can be easily filtered by the kidneys and not accumulate in any organs or in organism at all. Also, depending on the shape, you can see that accumulation in different organs differ. Right? And what about the coating surface chemistry? And in surface chemistry, here we use positively, negatively charged, not negatively charged, neutral, and positively charged particles. And you can see that with positively charged, there is higher accumulation in lungs, liver, and spleen than with negatively and neutral charge. So with that, I have one first poll question about coatings. So what if you look at coatings with 3 different charges, positively, negatively, and neutral? Which of those coatings will affect toxicity the most? And I know the answer that you answer says 50-50, and I can see why this can be a good guess. However, I want to show you like a little study results with oxide nanoparticles that we did. And this is TM image of oxide nanoparticles. And look, this is the core, the series atomic structure, and this is the coating on the top, kind of a lattice on the top. And that coating is dextran, which makes nanoparticles neutrally charged. We also use negatively charged VCM coating and positively charged shown in red. So everything about the particles, the size was exactly the same. The only difference was that the coating with different charges. So first we examined bioaccumulation using synchrotron images, synchrotron methods to look at accumulation. And we could see that there is stronger intensity, fluorescent intensity from Syria in the nematodes exposed to positively charged particles right here. And the reason why I can say that it's stronger, if you look at the scale here, right, it's different than the scale here. This goes from 0 to 40, and this intensity goes from 0 to 50. So, and indeed, when you look at the graph accumulations, that there was more Syria accumulated in the nematodes after exposure to positively charged particles in comparison with negatively and neutral. What about toxicity? Again, here in red line is the response to positively charged particles. These 2 lines are response to negatively and neutral particles. So it's definitely the increase in bioaccumulation also correlated with toxicity, and there was much higher mortality when nematodes were exposed to positively charged particles. So positive charged particles are in general more toxic, and we tested it with other particles as well, and then other negatively or neutral charges. The reason for it is because if you think about the membrane surface are negatively charged. So when you have positive charged particles, the attraction and binding is stronger to those membrane surfaces. The more particles we have on membrane surfaces, the more will be taken into. So to make the story a little bit more complicated, right, in addition to all the properties and different shape and sizes and coatings that particles have, there is also environmental transformations that they undergo too. So when the particles are present in the product, they're kind of like present in pristine shape form first. But when they're being released in the environment and enter wastewater, the environment that is very rich with sulfur and phosphorus or phosphates, then they are transformed. And this is example for silver particles. When silver particles are going into wastewater or in soil, they immediately start dissolving, right? And silver ions released. Silver ions are being oxidized but it doesn't stay oxidized for long, and very fast that silver oxide is transformed into silver sulfide. So it becomes sulfidized. When the particles become sulfidized, it obviously changes their solubility, and hence it would change their toxicity compared to pristine particles. So this is different environmental transformation. And obviously the sulfidation, for example, would result in change in size, right? But also there are maybe aggregation with other different ligands in the environments, like in soil environments. There can be some dissolution going on, and obviously it's change the particle, how much bioavailability occurs, and also the how particles interact with different like proteins, biomicromolecules present in the environments. Also, if you heard about Trojan horse, particle can be serving as carriers for other pollutants like heavy metals. Again, their transformation may result in the different ability, the way how they carry those pollutants. All of it eventually will affect toxicity. So with that, my full question number 2, how does transformation of silver nanoparticles, let's say from pristine silver to sulfidized particles, affect their toxicity? Will it increase or decrease? And the answer was, you guys answered that it's 50-50 in our live lecture. And I'll tell you a little bit result of another study with transformed silver nanoparticles in C. elegans and how it affected their reproduction. So what happened with reproductions here on the graph, you can see response, meaning average number of offspring, in response to silver ions, such as silver nitrate, in response to silver particles, which are pristine, and in response to sulfidized, which are transformed silver nanoparticles. And if you look at the scale, it's obvious that the most toxic are ions than particles, and sulfidized particles are about 4 times less toxic, right, than pristine particles. And we also measure toxicity. We also, in addition to measuring toxicity, we also measured dissolution, and dissolution decreased significantly in sulfidized or transformed particles, so they don't dissolve as easily. If you remember before, I showed you diagram where the silver ions were very toxic to aquatic organisms and plus to the nematodes. So that's what happens with decrease in dissolution. We also have decrease in toxicity because not as many toxic ions being released. And this is just to show you that silver particles are taken in different organs in the nematodes, but also in gonads. Nematodes have very large gonads. And if those particles are taken in the gonads and causing some toxicity in the gonads directly, that suggests that it can have effect on the subsequent generations. And our studies do confirm that there is effect of multi-generational toxicity. We look at the toxicity over 10 different generations. And what we observe that there is immediate decrease in reproduction as soon as in generation 2, right away, and it continues through 10 generations of continuous exposure in response to silver ions, silver nanoparticles, but not in response to transformed particles. The decreased toxicity in response to transformed particles is only observed in generation 10. Another thing that we did here, we also look at possibility to recover. So if you stop exposure after 5 generations and let them continue reproduce right through 5 more generations, then you have this one square here that is open, and that's a recovery. They don't recover when they're exposed to silver ions or pristine particles. they do recover when they're exposed to sulfidized particles. So multi-generational toxicity does occur in response to pristine particles. In response to sulfidized, it's much reduced. But again, we need to be careful because in the environment, it's not just sulfidized particles present by themselves, right? We have fungi, we have other bacteria, we have plant exudates, and those also able to remove that sulfidized coating and enhance dissolution of the particles and eventually you'll have toxicity. So they won't be transformed all the time. So with what, if I take into consideration what I said previously, and this is the diagram basically how when would be important to still incorporate safety in the design of your nanomaterial containing products. And what they recommend here in the blue line, that the cost at which you can still manage health and safety risk. And in red line, its ability to still impact and change the design to make it more safer. So when you select nanomaterials and then you start characterizing it, and then finally you do toxicity at different levels in vitro to in vivo, ability to influence it's before you go at mass scale operation of that nanomaterial containing product. So after you constructed the product, right, or the particles that will be used in the product better, and started doing toxicity, realize they're toxic, it's still possible to go back, redesign to see, to make it a little bit less toxic, right? Hopefully a lot of less toxic, and then continue to operation after that, after appropriate testing. So what do I mean by actually making it less toxic? What is it possible to do? Which are other safer alternatives that can be used in design of those particles? So here is just kind of the scheme, right? For example, we design particles, nanoparticles. A lot of those nanoparticles, maybe nanocomposites, have a different combination of different particles such as alloy, or it can be carbon materials with the metal particles. Okay, and those are particles that use, for example, in sensors, and then they go through toxicity testing in vitro, in vivo. If they're toxic, then why they're toxic? You can look at the different mechanism of action, right? Like why is it toxic? What can be changed to remove that specific toxicity pathway? But also you don't need to screen necessarily all the toxicity by yourself. It's because there are a lot of publications already available on different type of nanoparticles with different coatings. You might just need to be able to look through the literature and see what is known. There is also a lot of studies on different mechanisms that involve and responsible for the toxicity observed. So one of the design, for example, this is example with copper oxide nanoparticles, which have high dissolution rate. And to make it safer is to decrease the dissolution, right? And in this case, they were proposed to be doped with iron, and that decrease the dissolution. What else? There are also other alternatives that can be used, and this diagram shows different alternatives. For example, as we already discussed about positively charged particles, right, and bind tightly to the ligands, to the cell membranes. We want to decrease that binding force, so we can replace it with negatively charged particles, or there are maybe some ligands that can be used and that will allow also relax that binding. Even changing the shape of the particles may make the binding less strong. Another thing with unstable particles that are prone to dissolution, right? We need to figure out how to decrease the amount of dissolution of those toxic ions from the particles. And as I showed the example with copper oxide nanoparticles and iron coating on the top to decrease it. You can use basically different shell materials, or you can completely replace the toxic core element with a different particle. Gold even would not be dissolving, right? It's not dissolving one, but it might still be functioning the way you want it to function. Depends. Also, the shape can be changed, and that may also diminish the solution. This is really interesting suggestion. It's called chelation chemistry, where you chelate the surface. So that chelate is for silver ions that are being released, not silver ions, any ions released from the nanoparticle core immediately are chelated and are unavailable for being toxic. So that's pretty cool. Another thing that one of the main mechanism for nanoparticle toxicity, especially metals, is generation of reactive oxygen species. And there are also different alternatives that can be used to diminish production of reactive oxygen species. For example, here, if you use nano composites or like alloys, and one alloy is more toxic than another, one of the components in that alloy, like you use silver and gold, and silver is much more toxic to the environment, is possible to replace with something that is less toxic, less dissolvable, to improve that design. Another thing, it's possible to attach antioxidant ligands, right, which meant immediately will be responsible for decreasing generation of reactive oxygen species. So I also want to show to your attention two different databases where you can find actual information, some toxicity information for nanomaterials. It's open source applications. One is from Europe in Nanomapper, and another one is from US, is still being developed, and they're still adding materials to the database. It's in Oregon State University. And there you find information on nanomaterial biological interactions at different level of organizations. So highly recommend to look there. There will be also references to different studies that are presented in those databases. And finally, at the end, I just want to ask you one question. So with what you heard today, right, did your maybe opinion change? Do you think that nanomaterials need to be regulated separately from regular chemicals? Yes or no? And I know I was happy to see that everybody responded yes. So it's true, they do have unique properties and it's hard to regulate them, but they definitely are. So far, we don't have the strict regulations specific to nanomaterials. They are still being regulated as basically their counterparts as chemical characters. Like for silver, it would be based on silver ions, which is better than nothing. But for example, in US, you don't need even to report that nanomaterials are being used in the products. So I think they need to be regulated a little bit better and separately. With that, thank you so much. And if you have any questions and need to contact me, please don't hesitate to send me email, for example, and ask any questions. I'll be happy to respond.